I'm Dr Darren Roberts. I'm part of the Translational radiobiology group at the University of Manchester. I'm specifically in the Division of Cancer Sciences, and we're focusing on improving cancer outcomes for a variety of different patients, both in terms of chemotherapy and radiotherapy treatments. As part of translational radiobiology, we're interested in the effects or changes within the actual tumour itself and how that interacts with radiotherapy. We developed a prostate signature, and this was to predict benefit from a modified form of radiotherapy. We were interested in using a technology which would give us maximum amount of data on the old degraded samples. So in this study we investigated rna-seq using the Illumina platforms, and we also compared it against our current gold standard, which is the Tachman low density array. We needed a third technology to actually compare this against, and CLARIMS was the closest match we could find to the old Affymetrix array systems. We're quite agnostic for what technologies we use. We generally take the approach of using the correct technology for the correct sample. So we have looked at both array-based technologies. We've used NGS, we've used targeted NGS, we've used qPCR, and we've even used Quantigene. In our discovery phase and initial validation phases, we'll be looking for a technology which can cope with high numbers of samples. Because we're focused on radiobiology, quite often we'll be looking at hundreds, if not thousands, of samples, and we want it to be able to deal with old FFPE material. So the DNA and RNA tends to be quite degraded and difficult to get out. So at that stage we need a technology that deals with that type of material. We generally use CLARIMS within our discovery phase and early stage validation because it's an excellent technology for use with degraded RNA samples. In later stages of our projects, we move on to technologies such as qPCR. We have a phase 3 clinical trial ongoing using the Tachman low density array, for example, in a different tumour type. We looked at the RNA-seq data and found that basically there was very poor quality data there. We also looked at qPCR and we were having several genes dropping out. So we were quite relieved to see in the CLARIMS data that we had high quality gene expression data, and that has provided us with a database of the transcriptome of these patient samples for us to use going forward. And that's particularly important in this study because we were dealing with prostate samples, so we were dealing with really tiny pieces of tissue, usually in the order of 10 millimeters by about 1 millimeter, and we were taking a 4-micron section. What we consider now is the sample itself rather than the technology. CLARIMS arrays actually performed far better than we were expecting, particularly based on the initial RNA-seq results that we had received. If I was coming to this kind of study again, I would be looking at array-based technologies for early discovery with older samples. From there, I would then move on to targeted NGS or qPCR, whichever one's most appropriate for the clinical setting, to be able to progress these through to patient benefit.